thought of rewriting the code of your life or changing the fundamental chemistry of who we are? You sort of can. I'm sure you at least know what a DNA and gene are. Gene instruct our cells to make proteins, whereas DNA, the information manual for organisms, determines the way we look and function. It's made of 4 bases, A, C, T and G. With genome editing or CRISPR-Cas9, we can edit the code of our life. This genetic research transforming technology was an accidental discovery when a team of scientists was studying the war between bacteria and viruses. That is how bacteria protect themselves from viruses. They found out that bacteria have something called as CRISPR, an acronym for clustered regularly interspaced short palindromic repeats in their DNA, where they store tiny pieces of viral genome. So, when a virus enters into bacteria, the CRISPR-associated nuclease or Cas9, an enzyme acting as genetic or molecular scissors, will chop off a piece of viral DNA. This information is then stored between the CRISPR palindromic sequences. Thus, the bacteria makes a genetic memory so that it can avoid future infections. What are palindromic sequences? Palindrome is basically the unique DNA sequence that repeats itself in the bacterial genome. How cool, right? Summing it up, CRISPR, the naturally evolved defense mechanism of bacteria, whereas Cas9, the DNA cutting enzyme. Cas9 and gRNA or guide RNA acting like GPS tracker together form Cas9 complex. Scientists can design a short sequence of code within gRNA which matches with what you want to replace. Cas9 complex can now roam around DNA to find that matching bit of piece. Once found, Cas9 will unzip the DNA and gRNA will slip in to check if it matches. If it does, Cas9 will slip the DNA into 2. Imagine having drought and climate change resistant crops, more nutritious fruits and vegetables, fully biodegradable plastics, therapies for HIV, cancer, thalassemia, cystic fibrosis and many more. With CRISPR, this is not only imaginable but possible. For this breakthrough technology, scientists Jennifer Doudna and Emmanuel Charpentier won a Nobel Prize in 2020. Now, because editing our DNA has become almost as simple as editing a Word document, we should be well aware of its consequences. Imagine unintendedly getting rid of a huge chunk of your DNA or some catastrophic mutations which are beyond our imagination. Scientists foretell that we still have a long way to go before we can edit human embryo. Now, one more question which can come into your mind: why is CRISPR so revolutionary? It's because it's easy to perform, cheap and faster than normal interbreeding. Now that this technology is already here, seamlessly weaving in the fabric of physical reality, we need to regulate it and avoid its negative uses. It has an immense potential to cure and eradicate some of the world's biggest problems without hurting anyone, and we don't want to miss out on that.